So thank you, Vanessa, and thank you all. It's a pleasure to be part of this community, and I'm really impressed by the professionals, not only the clinical professionals, but the professionalism of the families. Because I've learned a lot, not only from the clinicians, from all our colleagues, researchers, but also from the families, and that's very, very important and very interesting. So I'm a professor, and I'm a basic scientist, and my main aim is to study the glycobiology and the immunology. So I'm leading the group called glycoimmunology. So if you think that the glycosylation is very complex, so immunology is also very complex. So the aim of this talk is to call your attention for the immune response that maybe, I'm sure it's not the main problem of our patients, but it can be a big problem in our patients. So I will just explain you how it works and how complex it can be. So will not enter into many details, so if you have other questions at the end, you can talk with me or you can contact me, so I will explain better. So I hope I will explain what is this about immune response, how it is affected by glycosylation, or how the deficiencies on glycosylation can affect our immune response. And I will give you a focus on dendritic cells because it's our focus, it's our main interest in terms of immunology. So we believe that if we learn more about how the immune response is modulated by glycosylation, we can get some clues to improve the health of our patients. But in the other sense, we can also understand better how the immune response acts, what are the exactly mechanisms, how it can be modulated. And it can give us also clues to other treatments for more common diseases like cancer. Okay. So what is this about the immune response? Or the immunity, whatever you call it, it's a system or a group of biological mechanisms which involve cells, molecules, and the main aim is to protect us against pathogens, against toxins, against pollution, parasites, bacteria, wherever. It's there all the time, it's protecting us. And so the role is to, we could divide it in three. Like to recognize. To recognize is very important because recognize means also to distinguish. So the immune response has to recognize what are the pathogens and to distinguish them from our cells, right? And also among the microbes, it has to distinguish what are the bad or the good guys, right? And it has to defend us only against the bad guys. And also the capacity to memorize. So we, whenever we are challenged with a microbe or a pathogen, we set up an immune response, and then in the second encounter, we'll be stronger. We'll have learned our immune response has been educated, so it will act more faster and better in the next encounter. That's what we expect, this memory of our immune system. And so there are many examples that you know about the immune response, and here I'm just highlighting the pathological issues, like an infection is when your immune system fights against the pathogen, allergy is when your immune system is exaggerated or overreacts against an innocuous molecule. The autoimmune disease is when your organism or your immune system is not able to distinguish very well what are the self or non-self cells, and so it fights against your own cells. And the vaccination is a means or an attempt to educate your immune system, to tell them or tell it what it should recognize, or in case of allergy, what it should tolerate. Right? But there are more examples of immune response that usually we don't think about, but immune response is there all the times, like cancer. In cancer, our immune system is able to fight cancer cells. So aberrant cells, it will be able to kill those cells. But at some point, you lose that capacity or tolerize them. Right? And other pathologies, there are many to describe, but just here the atherosclerosis or miscarriages, two completely different situations, but where the immune response is a disadvantage. Right? So, but as I said, the immune response is there all the time. So we are always being challenged with all of these toxins, virus, parasites, bacteria, and we even don't notice because it's working. Well, right? Our immune response is working, so we don't really notice when it's working. But imagine that you have a cut in your finger, so suddenly you'll get your finger red, warm, right? And it will start hurting. So that's your immune response being promptly acting. And we can distinguish our immune response into main systems. The innate immune system is very rapid. So you have cells like macrophages, dendritic cells that are already in the skin that will move and will try to eat these pathogens. This could be a bacteria, right? And you also have other cells that are in our bloodstream that have needs to exit the bloodstream and go to the site of the cut and to encounter bacteria to kill them and destroy them. So this is very fast, and this is what we call the innate immune system. But at the same time, there is mechanisms that will trigger another part of our immune system, which is the adaptive immune system. Here you have cells like lymphocytes or B cells, T cells, and their response is not immediate, is longer, but they will proliferate, and they are responsible to give us our immune memory, the capacity to memorize and to fight better in the next encounter with these pathogens. Okay? So again, just to distinguish these two types of immune responses, the innate and adaptive, but as I said, they are interconnected. And here you have cells like those that I mentioned, dendritic cells, macrophages, neutrophils, and which we also call them phagocytes because they have the capacity to eat, to engulf pathogens or bacteria, right? Or virus. And you also have other cells like natural killer cells, which are able to kill other cells whenever they are infected, like with the virus. Right? And here you have the B cells and T cells, which are also able to eliminate infected cells or eliminated pathogens. And you have different mechanisms which are responsible to give us our memory, immune memory. So all of these are very important. Okay? So what is about the glycosylation here? So if you remember in all these talks you have already heard about where is the glycosylation. Just to remember that our cell surface is this the lipid bilayer, and there is many proteins that are embedded in the cell surface. And at cell surface, we can say that basically all the proteins are glycosylated. So, and even the soluble secreted proteins like the antibodies that I mentioned, or the cytokines, which are very important for the immune system, are also glycosylated. And even the lipids are also glycosylated. So if you think about the immune system that has to act very rapidly, has to encounter, has to contact and send a message one to the others, they rely so much in these encounters, these cell-cell contacts, these receptor-ligand encounters. So if there is any difference on the glycosylation, this will be affected. Right? So I will just give you some examples how the glycans are important for the immune response. Here is an example how the glycans are important for the cell migration. Remember when I talked about the cut in the finger, where some of the leukocytes, like neutrophils that were in your bloodstream, needed to exit the bloodstream and go to the site of infection to stop the infection. So here, imagine that the cells are in our bloodstream in a very rapid, very fast. At some point, they have to be slowed down, to contact with the endothelium and start a process called transmigration to exit the bloodstream. And this first step is performed by interactions between proteins and glycans. And you have glycans that are expressed by the leukocytes, and they are responsible for what we call tetraenrolling. So the cells start contact with endothelium and tetraenrolling, and then that will give a more strong adhesion with endothelium and ultimately enables the transmigration. If there are some problems with these glycans expressed by leukocytes, the cells will not be able to transmigrate, so they will continue on our bloodstream. And that will give us some deficiencies that I will give an example like leukocyte adhesion deficiency. But there are many other examples. So now I will start focusing more on the sialic acids because that's what we have been interested with. And so the sialic acids, as the deficient on sialic acids is common in many CDG diseases. And the sialic acids are very important because they are there to shield us from pathogens. But we also have some pathogens that also recognize sialic acids, like the influenza virus. So you have the both sides of the coin. But during inflammation, remember, again, the cut in the finger, there is an increased expression of sialylated glycoproteins and the sialyl transferases and the sialic acids, because it's important to, again, to shield us from pathogens. But it's also important to give us some homeostasis on the cell leukocyte homeostasis, because you have a burst on your cells that proliferate, but then you needed them to put them at the normal level. You need the homeostasis. So the sialic acids are important for this process. And there are many other functions like immunoglobulin. So the glycosylation of immunoglobulins is also very important. And the presence of sialic acid is also very important for the functions of the immunoglobulins. Just one example that sialic acids can prevent the complement the position at cell surface. So the complement is one of the mechanisms of our innate immune defense. It's able to bind to pathogens and to destroy them very quickly. So, but not in our cells, because our cells have other mechanisms, and one of them is the presence of sialic acids. And so at some point, the presence of sialic acid prevents the complement deposition, and if there are some defects, we could be more prone to pathologies like autoimmune diseases. So what happened on the CDG cases? This is much more complex, and I just wanted to remember you that our cell surface is covered with many proteins that are glycosylated, and whenever there is a deficiency on a gene, in the case of CDG, and this gene has to do with the glycosylation. So there is a deficiency on an enzyme. This enzyme will transfer a sugar or remove a sugar. So that will affect many glycoproteins because there are many different proteins that can have the same sugar. Right? So there are many proteins that can be affected by only one mutation. And that means that different functions can be affected. So it will be always hard, especially on the immune system, to understand what are the mechanisms that are different, because many of them can be affected. So this is just to remember that, in fact, the clinical features of patients with CDG are many, and you know what are the most problematic. But one of that has been neglected is the immunology. So there have been several or few reports, not that many, about recurrent infections, mild inflammatory response, increased concentration on inflammatory cytokines, and there are probably more. So, but maybe we are not taking all the data, are not getting all the picture. Right? So why I think the immune response should be affected, according to my point of view, there are so many mechanisms involved in the glycans, immune mechanisms, like cell surface immune receptors are glycosylated, right? Many immune receptors are lectins. These lectins are proteins able to recognize glycans, right? And the many pathogens rely on the recognition of these glycans to invade our body. So if there are differences on our glycosylation, we have all this process affected. So now I will enter into what we have studied, just to give you some clues about our research. So we have been focused on dendritic cells because they have a key role on the immune response. So here you can see I hope you can see the dendritic cells. They have this name because they look like stars. And we have used a molecule, a lectin, actually, that recognizes glycans. In this case, it's sialate glycans, alpha-2,6 sialate glycans. So this SNA lectin can show you that there are many sialate glycans at cell surface of these dendritic cells. Okay. So why did I say that they are very important for our immune response? Because they are in the they are part of our innate immune response, but they are important to trigger the adaptive immune response. Okay? Here you can see the dendritic cells. They are phagocyting or eating a bacteria, and then it will digest this bacteria, and it will break it in small pieces, and it will present it at cell surface to T cells, and the T cells will be activated, and they will set up an immune response or not. That will depend on this contact. This contact is very important to tell the T cells fight it or not fight it, this antigen. And this is very important because the problems with these cells can give all the other problems I have mentioned already, like allergies or even autoimmune diseases. Right? But since they are so interesting, they are also being used as vaccines, cell vaccines. And this is very interesting because the principle is very, very simple. We might not know that much about dendritic cells, but since this treatment has been working, there are already some clinical trials with these vaccines. And there are already 2 vaccines that have already been approved. So the vaccine has been used against cancer so far. And the principle is very simple. So you get some precursors from the patients, cancer patients in this case. And the precursors can be monocytes. So you have these precursors in your blood because usually these cells, dendritic cells, are mostly in your tissues. So in order to get enough cells, you go to your precursors that are cells that can be differentiated into dendritic cells. And then you modulate it in vitro and give them tumor antigens, maturate them, and introduce into the patient. And hopefully it will set up an immune response against the cancer cells, and it will kill or vaccinate the patient against cancer cells. Okay? So again, dendritic cells have many pathways that are regulated by glycans. And here it's just to show you that there are some receptors like DCSINE and others, galactins that recognize glycans and are affected. And there are signalings that are sent to the cells whenever they recognize or not glycans. So what we did was to study these cells. We have been focused on human cells, human dendritic cells, and we look at the expression of sialate glycans. And here again is another type of staining of the cells, here in green. And in this case, it was for the sialyl with X, which is a glycan important for cell migration. And so our model for study has been the use of these human cells, as I mentioned, and we treat the cells with glycosidases like sialidases, or in some cases we also do some genetic engineering on these cells. And to confirm or to compare, we also have been studying dendritic cells from mice deficient in specific sialyl transferases. Okay? So we have tested many things on these cells regarding their functions. So we are interested in understanding how can it affect the functions. And these are the functions we have been studying. So migration of the dendritic cells, which is very important, activation of these cells, uptake of pathogens and tumor cells, and maturation and cytokine production. So what we found is that the effects on sialic acid in these cells influence the immunological function of these cells. It can give to higher expression of cytokines, higher ability to prime T cells, but at the same time also higher capacity to uptake bacteria and tumor cells, and higher maturation. So this was very important for us, and that gives us some ideas for what could we learn with these with cells, or the deficiency of sialic acid in these cells. But at the same point, for other type of sialic acid, because there are different types of sialic acids, it also can, the decrease of these sialic acids can also lead to decrease on the migration of these cells. So I will not enter into details, as I mentioned, but for us, we found that sialic acid could be a means to engineering, and it could modulate the immune function of these cells. And maybe we could use them for the vaccines to improve the capacity of the vaccines, because, as I said, they are already being used, but there are many things that we don't know, and they should be improved. And what we are now preparing is a preclinical trial where we are using this modulation of sialic acid on the dendritic cells in order to improve, because as I mentioned, the deficiency of sialic acids give these cells the ability to trigger better T cells. And the decrease on sialic acids is actually physiological for dendritic cells, because there are many sialidases that are secreted, or even during the maturation, there are also decreased levels of sialic acids. So actually, we are mimicking something that physiological already exists, but we are forcing. And we can use that knowledge to think about a therapy for a more common disease like cancer. But what this data also suggests for the CDG patients. So the defects on the sialation are also observed in many CDG patients. It is already been here mentioned. And these specific sialate defects on the dendritic cells that we also observe in our experiments could explain why we, in some patients, we see some increased inflammatory response. Because as I mentioned in our model, the deficiency of sialic acids leads to increased inflammatory response, increased expression of cytokines. And in another case, also the effects on sialic acids lead to decreased migration that I also mentioned. And this may explain also cases with increased propensity for the infection, since the cells are not able to migrate due to this deficient on cell migration. Okay? So the take-home message here is that the immune response can act in many, many ways, and it is associated with pathologic conditions, not only those that we have been heard, but many other pathologic conditions. And the glycosylation is very important for the immune response. So the more we learn about the glycans, how it affects the immune response, we can have some clues that will help to improve the health of our patients, but also other cues for other more common diseases. And I will take this opportunity also to mention that we are also working on the e-learning course that we hope it will be able or available in the beginning of next year. And it is about glycoscience. So I also invite you, if you have ideas or if you would like to participate in this course, to give some lectures, please come to me, and I will be happy to take all the things necessary to record, or I'm sure, even with short money, we'll get some idea that will make this the communication and also the spreading of the glycobiology. I think it's something that we should improve. And also, about this course, if for the patients, even for the families, if you think there are something that you'd like to know more about glycosylation, it will be interesting to know if you are willing to have like a e-learning course or a basic course to inform you better about glycosylation. I think that will be interesting to use this kind of platform to also to inform you about this type of diseases and other type of subjects that you think that will be interesting. Right? So this is my course, my group, and we have many PhD students and also postdocs, and we have 2 clinicians, and we are always receiving some medical students in our group. Okay, and so thank you.